Hi, everybody. Welcome to week 3. This week is going to be pretty much an informational type week. We will discuss some techniques that are being done in the microlab for different testing, so different principles of testing. This is also a kind of a crossover with your immunology course, so you'll learn a lot more about some of these techniques in immunology. So we kind of keep it pretty informational here. Mainly because when you get to clinicals, you'll really get to play with some of this instrumentation there, especially. So we have 2 different PowerPoints to go over. The first we'll start with is called Immunodiagnostics of Infectious Diseases. All right, so in some of our test methodology, there is agglutination reactions and precipitation reactions. And again, this is heavily discussed in immunology. So precipitation and agglutination are both a visible result of when antigens and antibodies come together. So the difference would be precipitation is from soluble components. You notice the word soluble there is underlined. Agglutination is the clumping or aggregation of particles that have antigens on their surface by antibodies, and then they start to link and bridge to kind of make it more visible, a clump. You know, obviously we cannot see an antigen and an antibody coming together. That is something that we can't see with our eye. But when they start to do it enough and they link and bridge together, then we can see it as a clump or a precipitate. So main difference here is just precipitation is more soluble components versus agglutination, but both have antigens and antibodies coming together, bridging together to make a visible clumping precipitation. So if we kind of look at if we look at a diagram here, so your antibody is the Y shape, and then say your antigen is the big round green guy. You can see like they'll start to bind them, but we can't see them but then you can start to see how they can kind of connect together, and you get them connected enough together, then we can see it with the eye. All right, so there is different techniques called latex agglutination where they bind latex beads to the antibodies in that test. And so they're looking for, again, a specific antigen. And so what I should preface by saying is when we're looking for an antigen, a lot of times we're using a test that's like we're deciding if somebody has infectious mono or strep or influenza, had kits like this, things like that. So we're looking for a specific antigen, a specific viral bacterial antigen that we're trying to detect. And so they will have antibodies in that test kit. And in this case, they have latex beads on them. And we add the patient sample. And if that antigen is there for that antibody, they bind together. And so the latex beads here just help create more of a visible aggregate if it's present. Pregnancy tests, are, as it says at the bottom, there is another great example of when this is used. So once they bind together in the case of pregnancy, a lot of times they'll make a colored line or like a plus symbol, or, you know, however the test is on that. But again, it'll become visible. So if we look at with the latex beads, again, they attach these latex beads to the antibodies, and then if the antigens there are, they connect. But the latex beads just really help form that agglutination because they're bigger, so we have a bigger bridging happening with it. So here's kind of how it might look in like on a slide or a tube version as well. Again, in kits, it might be a colored line that's helping to form, things like that. There is direct bacterial agglutination. So in this case, as the name implies, we're looking for specific bacteria, pathogens. So we are looking for these antibodies directed against those pathogens. So you can take a thick suspension of bacteria, and then those antibodies will bind to them. And then again, you have agglutination occur. So different things that will influence our agglutination. Basically, it's 2 stages. It's the sensitization. All that means is when an antigen and an antibody come together, the attachment of the antigen to the antibody, that's sensitization. And it'll depend on the pH, the temperature, incubation. There's like different factors that go into play with that sensitization step. But overall, that step is just the actual coming together of the antigen and antibody. And then the lattice formation is where they bridge together. They link together so that we can visibly see the agglutination. So that's the lattice formation. There is different cells that could become involved. We do have red cells. When you take blood bank, we will learn you can type people's blood based off agglutination reactions with the red blood cells. So there definitely is agglutination kind of stuff happening there as well in blood banking. that involves cells. So just kind of 2 stages overall. Again, this is heavily discussed in immunology. So that's the sensitization. So these antibodies and the antigens are going to join together, and then they lattice. So you can see that they are linking all together to become a bigger clump. Okay, flocculation tests are dealing with precipitation. So that's soluble component. So instead of calling it agglutination, it's precipitation. So this is flocculation test. Probably the main ones that we deal with would be syphilis testing. The VDRL and the RPR, 2 different screening tests that use precipitation testing methods in them. So these are screening methods for syphilis. If that was positive, then we would reflex over it and we would confirm the syphilis with a different method. But these are just quick screening tools to say yay or nay. If it's yay, if it's positive, then we would confirm it first. So there are types of precipitation tests. There's what we call radial immunodiffusion, rocket electrophoresis, and immunoelectrophoresis. So we're not going to get into depth on these here too much. You will hear the term immunoassay quite a bit, especially in regards to there's certain parts of chemistry you might hear this in, immunology. Immunology is kind of one of those areas that's spread a little bit throughout all of the lab because you have different immunology methodology happening in most of the lab. An immunoassay is just a test that involves antibodies. So anytime that I see the word immuno in the title of something, I know an antibody is most likely involved. So you can label those antibodies with different dyes to form different colors, different enzymes, different radioisotopes to kind of like fluoresce or put out a color change, or, things like that. That way it kind of helps us know, oh, something happened here. The antibody was found its match. So they will label these immunoglobulins, just a fancy term for antibodies. Again, to increase the sensitivity of the method, to be able to see that, yes, truly it's positive, something was here. So you can have, again, enzyme immunoassay, meaning the label is an enzyme, radioimmunoassay, meaning you have a radioisotope label, or fluorescent antibody assay. In that case, they're using a fluorescent type dye that's going to fluoresce at the end of it. So here's a look at like an enzyme immunoassay. This one was a strep test. We have transformed, most labs have transformed into using molecular techniques now for strep, but a lot of small clinic labs especially might still do a strep test. They were simple to purchase, they're easy to use. A lot of times you can get ones that are waived, so almost anybody can run it if they've been trained on it. So it is an easy thing to perform just based off a swab of the throat. So again, they're looking for the antigen antibody, and then they do a color change. And in this case, enzyme immunoassay. So they have an enzyme labeled on there. All right, so this is just kind of showing again that enzyme immunoassay reaction, what's happening. They have an antigen on the plate, the antibody binds to it, and then a second enzyme labeled antibody comes along and sees, oh, they're joined together, I'll bind there as well. And then that enzyme is converted to a color. So it goes from a clear non-reaction to all of a sudden a pink color here, saying, hey, it's pink, it's positive. So that told us it was positive. So if you go back up, you can see that letter A is a negative. There's not much happening in that circle sample well. B formed a nice colored pink triangle, and that's the enzyme immunoassay part. Like that's displaying, yep, we have a positive strep here. Again, same, very similar method with direct fluorescent antibody. As in this case, you're using a fluorescent labeled antibody. So it's, of course, going to fluoresce when it binds together. And then you can see that with a fluorescent microscope as well. Indirect fluorescent antibody, same premise, just an extra step kind of added on. I'm not going to get in depth on this because this, again, isn't something we are testing on in our micro class. It's going to be more immunology that you will get to know these things and get tested on there. So this is just showing the direct versus indirect methods of that fluorescent test. So again, a lot of information. Overall, we use these immunology-based methods to look for different viruses, bacteria. A lot of times they're in kits, and kits can be cheaper, easy to use. They produce a color reaction or something to tell us it's positive. But we can also see immunoassays done, like I said, in chemistry on your big chemistry analyzers when they test for different things like thyroid testing or stuff like that. They're looking for a lot of times antibodies like in there. So that's where the immuno part comes in as well there. So Lots of different uses across the lab. So now we'll flip over to our other PowerPoint, and this one is pretty brief. This is about molecular techniques. As you can see, it's very minimal slides here. Molecular has become huge. It is the way that the lab is going, and it's going to keep moving to molecular. So when you hear of anything being a molecular technique, that means DNA, RNA is involved. We are looking for DNA of something. We are looking for maybe RNA of something, but molecular is all about nucleic acids. And so it is, of course, much more sensitive to determine if something is there based on DNA. If we can find the DNA, that is a much more guaranteed result if you find the DNA. Plus, molecular has really made our micro testing a lot quicker. Like traditional micro testing, which what you guys are doing in the student lab, takes a long time. It's like a 2-day, 3-day process to finish the result to the doctor. So if you get a culture in, you have to first grow it. Then you're going to gram stain. If you think you have something there, you perform the testing. You set up to finish identifying it through that testing. And then you also have to do antibiotic susceptibility testing, like what antibiotics can be used for this one. And so that's a 2 to 3 day process by traditional methods. So it was never a quick, fast thing in micro to have anything ASAP or stat. It just doesn't get done like that. We have to wait for the things to grow and then wait to play with them before we can even get there. So molecular has sped up that entire process. We can now put something in a molecular analyzer and have a result within a couple hours because all we're looking for is DNA. So it is a huge, it's a pro for patient care. Like your patient care can be so much better because we can really know what's happening within a couple hours, target that care, that antibiotic treatment, whatever it might be, much more rapidly as a result. So unfortunately though, for student labs, we don't really do molecular techniques in student labs because they're expensive and they have a lot of setup things and you can't contaminate and all this stuff. There's a lot that goes into them, but they're just expensive to have molecular equipment. This is where when you guys get to clinicals, or even when you begin working in the lab, you will get trained in on that instrumentation and learn more about it, how it works, what it can and can't do. And it's not just micro that has molecular. Molecular is, you'll see it in a lot of the areas of lab now is being done for many different things. All right, so with that whole setup, some things about molecular nucleic acid hybridization. Like I said, molecular is all about DNA, RNA. So when we say nucleic acid hybridization, it's just the nucleotides forming bonds of single strands of DNA and or RNA. So they're going to form double-stranded DNA, RNA, or RNA, whatever it might be testing. They're coming together, they're forming bonds between those single strands. That's part of the nucleic acid hybridization. So one strand is going to be your target strand, and that's what we're looking for with the molecular technique. The other strand is the probe and is labeled so we can detect it. So when it finds its bond, when it finds its other strand it likes, then it will kind of give off, like that label will give off a signal like, hey, we bound to our target strand. We found that target strand you were looking for. It's positive. So if it's, say, looking for tuberculosis and it finds, the tuberculosis DNA that we were looking for, that probe that's labeled will signal it. So we know, oh, that DNA was present. So that means that tuberculosis bacteria was there. It was present. Solid support hybridization was something we called blotting. They had a southern blot, northern blot, western blot. It depends on what they were looking for. They were either looking for DNA in the southern blot, they were looking for RNA in the sample in the northern blot, or in the western blot, they were looking for protein. Western blot used to be used heavily as a confirmatory test for HIV. It is no longer the main confirmatory test for HIV, but it was for years. It was looking for a specific protein from the HIV virus to confirm that it was there. Like we would screen people for HIV with other methods. And then if it was positive, it would reflex to a Western blot. And that Western blot would look for a specific protein that's found with that virus to say, yes, the HIV virus is there. I don't believe for the most part they use Western blot anymore for that confirmatory testing. I feel like they found other things, but that's what I used to remember it from heavily. But it just shows you it's other ways to look for, again, to confirm, to see if some virus or bacteria is present by either looking for the DNA of it, the RNA of it, or the protein from it. So again, their target nucleic acid, they have a labeled probe that's going to go and try to bind with it if it's there, and then a color reaction, a luminescence, something's going to happen to signal that it was there. So a lot of times our STI testing, like chlamydia, gonorrhea, is probably the most common STI testing that we send, that we collect, and that will get tested in a lab or sent out to a reference lab, however your hospital or clinic does it. Chlamydia, gonorrhea is the easiest ones to test for, plus they're the most common. Chlamydia is very common. So this is all pretty much done only based on molecular techniques now. You can't, gonorrhea you can grow on an agar plate, and we'll learn that in a few weeks. You cannot grow chlamydia on an agar plate. It doesn't happen like that. It is not easy to play with like our other bacteria. Gonorrhea you can, but chlamydia you can't. But it's much simpler to do a swab of their genital area and then send it off, and you're looking for the DNA of the chlamydia or the DNA of the gonorrhea to determine if it's a positive test for them. So they use probe-based assays for those. That is very common. I don't think anybody really does chlamydia, gonorrhea any other way besides based on the DNA. And again, it's a quicker result then as well, so that they can get treatment for the chlamydia or gonorrhea. Polymerase chain reaction, PCR, this should be something that you have heard about in the past, whether it's through a basic biology course, basic chemistry course, just at some point you probably have heard the term PCR, and that is where we just amplify DNA over and over and over again. So we take DNA and we need to make a ton of copies of it. And so like in this case, it says over 40 different cycles trying to copy it again and again. So we have a ton of DNA to play with in the sample. And so they will basically take your target DNA, separate the double strands of it into singular strands, join it with a primer, and then make new strands of that DNA. So that is all what's occurring in PCR. So again, it's still a molecular technique because it's playing with DNA. So PCR and micro, different reasons we would use this. Again, just to diagnose different bacteria, viruses, even fungi or parasites. There is a system called the Cepheid Gene Expert. That is a very heavily used molecular analyzer that is found out there. This has been used for all sorts of stuff. Look at the goodness it can do down here. So MRSA, we will discuss in a couple weeks. But if you've never heard of it, just hang on. You will in a couple weeks. C. diff is a common one. You get to know about C. diff real quick when you work a hospital, nursing home, clinic. Norovirus, right now, I think there is a norovirus happening as I make this recording. That is an outbreak that's occurring. It is a nasty diarrheal GI tract viral infection. I've actually heard it's both vomiting and diarrhea, and you feel like death for a few days. And it's easily passed in the community, very easily passed. So norovirus, we can detect viruses. You are just really tricky buggers to play with. So you can't grow them. They don't grow. They're actually not even living things. But we can look for the DNA of that norovirus to determine if somebody has it. And you can get that result again in just a couple hours, within a couple hours, so that they know, and the doctors can start to figure out if they need certain treatments, or like telling them to isolate, so that they don't pass it to the rest of your family or something. Ebola, we've heard of big bad Ebola, tuberculosis. The thing with tuberculosis is we will learn a little bit more on this next quarter in Micro 2, but it is a bacterial pathogen, but it is a slow growing one. In fact, it takes a couple weeks if you want to grow it. It does not grow even in 2 days. It takes a long time. Well, to treat somebody, you can probably treat them based on their symptoms and get an idea what's going on. But if you want to actually have a confirmed diagnosis, plus try to figure out antibiotics and stuff, you don't have weeks. Like, you want a diagnosis for that provider within a couple hours. So you can do that based on DNA and PCR techniques. All right, so the future, it's just going to keep getting to be molecular. Everything is going that way. It's getting to be cheaper as they figure out this technology and they fine tune it. Just like any brand new technology, when brand new technology comes out, at first it's more expensive and it's complicated. And then as they get familiar with it and they figure out things about it, it gets a little easier to use and cheaper because they're getting efficient with it. So it will just get more efficient, more accurate constantly as we're playing with this techniques. And it's just going to become more and more in that. So Micro is becoming really a different world. It's going away from all the traditional bench testing that we do in the student lab, which is fun. It's really fun to be so hands-on, but it's going to just analyzers running it, but for the better for the patients. But it's tough because, you know, those analyzers aren't perfect. You have to understand that when they give you a result, does that result make sense? You can't always trust the analyzer. You cannot say, I believe in this result every single time it gives it to you. You have to have a critical eye with it still. You have to assess, does that make sense? And then if it doesn't, you might have to go back to the traditional method of, okay, let's confirm this some other ways. Or your analyzer breaks. That happens too. Machines break. So you have to always be able to still know the traditional testing and the results that should go along with it. All right, so that was just a bunch of information for this week. Again, I don't really test you on any of this on a quiz or exam just because it's hard to really delve into it without being able to play with the instrumentation. So that will be coming in your future, but it is pretty cool what's out there and it's amazing. So, all right, that is it for this week. Have a good rest of your week.